I'm joined today by Jeremy Wheeler, who is the CEO of Biocaptiva, a revolutionary medical device company with the potential to transform liquid biopsy testing for cancer management. And he's a life sciences industry veteran, having worked with Medstone, Applied Neurodiagnostics, Aircraft Medical, and others in leadership roles. And he's been running Biocaptiva since January 2021. Welcome, Jeremy.
Thank you. Nice to be here.
Tell me about your background and upbringing. Where was Jeremy, formed?
Sure. Okay. So, I'm an Essex boy. I am the son of an electronics engineer. I spent quite a lot of my childhood growing up in Iran. And then came back to Essex. Coming back to an Essex comprehensive, that was a bit of an eye-opener, having been in a private school in Iran. But anyway, so I went on to become an electronics engineer by training. So that was, that was the basics. So always electronics, originally with Marconi's, and then with a Swiss company called Zumberg, which was non-contact measurement using scanned lasers and ultrasonics and things like that. And I worked for them throughout the UK, in Switzerland, and in the US, as it were. And that was sort of my introduction to sort of small companies and many hats, shall we say.
What was it that got you interested in life sciences then? Where was the trigger point there?
Sure. So I was approached by a company called Krios International, sort of well known within the Scottish medical device startup circle. And they were looking for a global service manager or some such startup type title. I came to talk to them. It seemed like an interesting challenge. The technology was extremely interesting. This was high frequency, high voltage X-ray generation, power generation for X-rays, I should say. And that was my first introduction to medical devices. So what I was in was a startup medical device company full of very empowered and very intelligent people working on a global scale. So I really enjoyed the cutting edge engineering with the regulatory framework of medical devices, with the global element to it, and also the element of even when you're having a really bad day, you're doing it for a good reason, as it were. It's this whole element of trying to make people's lives a little bit better. That's a good reason to go to work every day.
What was it that basically triggered your transition from, a more kind of broader base kind of electronics environment into medical devices? So you had that opportunity through Krios, but then what kept you there? Was it
Sure.
that purpose?
Yeah. So when you look at my background and you look at my personality type, I'm not a researcher by nature. I enjoy application. I enjoy process. So what I really like is bringing lots of technology and ideas and structure and process to a company to make it work. Shall we say, I'm not a good enough engineer to engineer a fabulous product. However, if you change the scope a little bit and you say, well, okay, a startup company is a product, I can bring all of those different bits of experience and all those different bits of knowledge to make the company the product. And the fact that I'm really quite good at translating what different skill sets of people mean to other skill sets of people makes me sit quite nicely in the middle. And I enjoy knowing what I need to know in order to get the result, and then learning new things. So that's what's kept me involved, and particularly in startups, because there's always a new tech challenge. There's always something new to learn. You never get bored.
Yeah. I mean, it sounds like the engineer's mind, though, is still very much in play with that dovetailing, problem-solving kind of approach to, resolving issues ongoing.
Exactly. Whilst I'm not necessarily a very good engineer, I am to my core an engineer. It's how I look at the world. And the other thing as well is that I like and believe in manufacturing. Designing things, turning, let's call it raw materials, into other things, to make a product, to export it. That just sits very, very well with who I am as a person.
So just like pragmatic outcomes over theoretical pontification, as it were.
Yes. I save that for the pub and the second or third beer.
What was the big idea then behind Biocaptiva? And
so the genesis for Biocaptiva was from a professor at the University of Edinburgh, Tim Aitman, who is a geneticist. So to give you the big picture, liquid biopsy is an area that has been gaining traction for the last 10 years for the monitoring and management of cancer. So what it's based, liquid biopsy is simply taking a liquid and looking within that liquid for markers that relate to disease states. So it could be urine, it can be saliva, it can be blood, all sorts of different things. But one of the principal areas has been looking at cell-free DNA within blood, from a blood draw. And what cell-free DNA is that all your cells in your body die and regenerate. As the cell dies, it releases fragments of DNA as a waste product. It just sheds blood, rather like skin sloughs off your epidermis, as it were, little fragments come away. Yep. And this is a waste product, and it goes into primarily the bloodstream. So cell-free DNA are just these fragments. Now, healthy cells die, but also cancer tumor cells die as well. So you have not only fragments from healthy cells, you have fragments from cancerous cells.
Right.
And this is a subset of cell-free DNA called circulating tumor DNA. And within these circulating tumor DNA, so ctDNA, this has biomarkers that relate to the cancer. So that's the exciting thing about it. So what it means is that you can take a syringe full of blood and you can look for biomarkers within that blood that relate to cancers. And the reason that may be useful is that if you cannot get to a tumor to see a physical biopsy, or if you can't see it, or if it's very spread out, you can possibly detect things through the blood. So this is great. This is a very, very exciting area. Very exciting area. However, there is a fundamental problem, and that is that not all cancers are created equal. So different types of cancer shed different amounts of ctDNA. So if you have something like lung cancer, it produces a lot of ctDNA. If you have something like a brain cancer, it produces very, very little. And
Right.
equally, the bigger the tumor, logically, the more ctDNA.
Yep.
The effect of this is that a syringe full of blood is a finite supply of blood. So if you have low shedding or small tumors, there's not enough of this ctDNA in order to reliably determine the cancer. So that's
Right.
the fundamental problem, and this is true across all of liquid biopsy with cell-free DNA. So Tim's idea was that, well, if you use apheresis, so imagine apheresis is like dialysis, whereby you have a cannula, so a needle in one peripheral vein. You take the blood off, you separate the blood out into plasma and into the other component parts, and then you recombine it and you put it back in another peripheral vein. So it gives you a closed loop system, as it were. So if you imagine, if you can develop a device that is a bit like an inline fuel filter for a car, and you put it on the plasma line to collect cell-free DNA, then you've basically got an unlimited supply of blood. So you've got an infinite size of blood draw.
Right.
And this was the idea for the Biocaptis, and this was what we spun out of the university.
Right.
And the clever piece within that is that the filter, the Biocaptis, if you imagine it as a can with a high surface area substrate, it's like a sponge inside,
Yep.
and it's covered with a polymer, okay? And the polymer physically grabs the cell-free DNA as it goes past. That's the clever bit, without affecting the blood, so it's all biocompatible and this sort of thing. And then at the end of the apheresis run, 30 to 40 minutes, something like that, you can take this canister, you can take it to the lab, and then you can wash the cell-free DNA off, concentrate it down, and then I can hand you, the diagnostic company or the research company, 100 times more cell-free DNA than you will otherwise get from a single blood draw. So it's one of those things whereby it was exciting because the problem is very defined, it's very real. You know, you don't have to convince anybody that it is or isn't a problem. Everyone knows it's a problem.
Yep.
The solution at a layman's perspective is straightforward, and the technology is incredibly clever and patentable. So that's a really nice sort of set of things to take forward to the market. And of course, its direct effect on, I don't want to use the word humanity, but on cancer patients is huge, be it in order to give the oncologist more data to better determine the therapy.
Yep.
Or another interesting one is monitoring the disease state, especially when it gets into an area called MRD, minimal residual disease. And that is when there is so little that the oncologist may turn around to you and say, I have great news for you. You're in remission, we think. And you go, that's amazing. That's fantastic. I don't have cancer. But at the back of your mind, you're going, oh, but do I? And with these sorts of technologies, you've got much more certainty potentially about whether or not the cancer is really in remission. And if it does come back, catching it much earlier.
Yeah.
So this was the principle, this was the idea. that we spun out of the university to be the Biocaptives.
Very good.
And
Very good.
as we've gone out and we've talked to the market, what they have said is, well, okay, that's exciting, but this polymer stuff of yours, what else can it coat? What else can it do? And, you know, in my innocence, I've said, well, what do you mean by that? And they said, well, can you coat, for instance, magnetic beads, use it in an ex vivo scenario? I talked to our scientists. Yes, we can. Could it work with pleural fluid, so lung fluid? Yes, it can. Can it work with urine? Yes, it can. Can it work with whole blood? Yes, it can. And what this means as well is that besides its use in vivo with apheresis, it also means it has many very exciting applications ex vivo, which greatly broadens out the offering that we originally thought was exciting enough to work towards the market.
Yeah.
So that's been fantastic. So
Right.
the utility of our polymers is far greater than we thought. So what it's meant is that we've moved from being a purely device company to actually a platform technology company.
Right.
That's a
Wonderful.
bit of a long-winded answer. My apologies.
No, and it's no. I mean, it's very interesting because there's a whole journey there, isn't there? And
There really
you know,
is, yeah.
I love that kind of. I don't know. You start in one place and end up in another, and the
Yeah.
the amount of times I hear that actually is remarkable. You know, I think nobody ends up doing exactly what they thought they were going to be doing necessarily, but you know, it's all the more rich a journey for that. You know, so
It's all part of the it's all part of the startup lifestyle and experience, and part of the reason we do it.
what then has been your biggest challenge in the early days of Biocaptiva?
Well, probably like most of the people you talk to, I will say the pandemic, which it kind of was and it wasn't. So I agreed to take this journey. I agreed with the university to take this on and spin it out. And we went into lockdown 3 weeks later. So that was
Timing.
interesting. And it also meant that some potential funding that was in place disappeared. So it meant that I had to spent the lockdown writing the prospectus, making contacts, raising money, building a board, and we went through that whole journey, spun the company out, and had our first board meeting all virtually. So whilst other people were perhaps taking a fairly relaxed time during the pandemic, I was working my little hiney off. The good news was, though, I don't know if you remember this, but during that time, you could get access to people that you couldn't normally get access to. So it meant that I was able to reach out to some people for our board who perhaps wouldn't have first considered of, shall we say, getting involved with the likes of us at our very early stage. So that was really good. So that was the first one. The next one, really, besides all the normal trials and tribulations, is that setting up clinical trials is hard.
Yeah.
And I would say with all of the transitions that have been going through with the pandemic and everything else, it has made it harder. And also we've been changing from the Medical Device Directive, the MDD, to the MDR. There's been changes within the FDA. All of these have been challenging and added time. On the technical and the operational side, the business has gone superbly well. But on some of these elements, it's been challenging.
Tough one. Yeah. Well, I mean, I suppose none of it can ever be done without some sort of challenges, and you've been able to bring a lot of experience of relatively early stage businesses to making it happen. If it was all just a bit too easy, it might not have been.
There is an element that there are, because my team is relatively young. There is an element that we come across situations where I can look at it and go, okay, the first time I experienced something like this was 20 years ago, so I know what not to do. Let's try this and that, and that does smooth things out a little bit, and also, should we say, reduces some of the panic that can circulate within startup companies occasionally.
Set in for no apparent reason, perhaps.
Yeah.
But, everybody will feel it. What are you doing to commercialize the business?
Yeah, absolutely. So at this moment in time, in fact, we have I think it's next week, we have a new head of BD joining us. And he comes to us from within the diagnostic industry, which is great. So we're doing a couple of things. We are not the consumer-facing part of it. We are the cog that makes other wheels turn, as it were. So I spent a lot of time talking to CROs, talking to labs, talking to diagnostic companies, also talking to patient groups and things like that about the use of apheresis. Over the last 2-3 years, really just talking about our technology. Because we're quite defined in what we do, it means that identifying partners is relatively straightforward, shall we say, and trying to determine what are the questions they're asking, what is the evidence they're looking for.
Yep.
And we're now getting to that point where we have the evidence, as it were, and we're really now making the push to find partners. So that being, for the Biocaptis, we are actively looking for an apheresis manufacturing partner, because we sit well together with them, as it were, and it would be better to align ourselves with a single player rather than try and be all things to all people. But also talk to some of the, let's say, call them collection companies, or the pre-analytic companies, the extraction companies, and that is to position ourselves as a partner to offer coatings and things like that, of the polymers, trying to reinvent some of the. We don't want to be a better mousetrap. Let's say we want to go to the mousetrap manufacturers and say, okay, we can give you a technological advantage. So let us leverage your market knowledge. Let us leverage your marketing ability. Let's leverage your size and your funds in order to do something very special here. Because the main thing is that we want to see this technology is sufficiently important that we need to see it get to as many people as possible. This really is something I look at, and when I first got involved in it, going, no, this is properly important. It's got to be done. It's got to be out there, because it's going to make a genuine difference, not a niche difference. It's going to make a genuine difference.
Yeah. Great purpose. What's that journey been like with investors and their involvement, and how you've kind of perhaps approached some of the challenges around, raising funding and working with people who, hold purse strings, etc.?
Yeah. It's been pretty good for several reasons. Obviously, when you look at the makeup of our board is made up of people been there, seen it, done it. Of course, that helps. It gives you a level of credibility when you go and talk to people. The other thing is that the thesis that we put forward, as it were, really chimes with people on a number of different levels. Be it if you're looking to invest and make money, it's like cancer detection, big market, important market, lots of interest. Be it the human level, we virtually all know somebody who's been on that journey. Be it explaining it to a lay person or a semi-lay person, as it were. These are all things that fit very nicely. And then the other thing, of course, is that the Scottish ecosystem for funding of startups is extremely good. It's pretty easy to get to know people. I know that in Scotland we don't have the deepest pockets in the world, but that getting you started is really, really good. Really good. It also helped that our lead investor is Archangels, who everybody of course knows, and Sarah Hardy, their investment director, she knows me from Aircraft Medical right from the start. So that didn't do any harm, as it were, either. It's just like, oh yeah, I've seen him on a journey that worked out. So that's always quite useful. And of course, Scottish Enterprise, as ever, have been great supporting it. So it's been relatively straightforward. Raising money is always hard. It's never fun. There's always, I wouldn't say arguments, but there's always disagreements in how much people want to give, what's the percentage of the company. But our investors have been unbelievably supportive.
Great. And, obviously across a period of tumult, let alone anything else, in terms of COVID-19 and I suppose kind of the great surge in interest in kind of life science, followed by the drawing back into horns, etc. So it's nice to know that you're supported and actually so positive about the Scottish ecosystem, because I think you're right. It's nice that it is a relatively straightforward process to get started, at least. Not easy, because it never is, but
no. But it is get started though. I will be singing, when we go for the next raise, which will probably be a big raise, it will probably be a little bit bigger than the Scottish market can handle on its own, which is a bit disappointing. Scaling is always a problem in Scotland. It has been ever since I've been, doing startups in Scotland, which is 30 years. I would like to see it different, but we can but keep trying, as it were, and we can but keep having our successes. So no, it's a good place to start.
What are the biggest distractions in the way of getting a company like Biocaptiva up and running?
Well, very straightforward one for us. My concept is laboratory space.
Right. Okay, that's not an uncommon view.
Yeah, laboratory space, especially if you are Edinburgh centric, is it's incredibly difficult. It's incredibly difficult. It's I could see, this isn't an intention for us, but if we had a pull from another direction, I can see how Edinburgh would lose companies because of lack of laboratory space. I'm sure if I wanted 3,000 square feet of laboratory space or 6,000, I'm sure I could get that in a heartbeat. But the reality is the university have been great in that we have space within the University of Edinburgh. That's been fantastic. But we're getting to the point now where we need our own facilities, not huge, and they are not available. And the way that the available spaces are structured, as far as access and things like that, isn't very coherent to, let's say, a pragmatist like me. It's very, very disappointing, and it is, shall we say, the biggest unnecessary pain that I have.
Yeah, I can imagine. And, that is a view echoed, throughout the community, I think.
Yeah.
You would have thought that it was a relatively easy fix, but doesn't seem to have, got onto the horizon or got onto the radar, I should say.
Yeah.
The powers to be.
If I had a magic wand, I would take some of the larger spaces, subdivide them into 1,000 square foot, a couple of 1,000 square foot, and I think that you would you'd have them built very, very quickly, and you'd have a much happier band of startups, shall we say?
There you go. Good advice, I think, for whoever's listening. You had an opportunity to go back and give yourself advice on the journey that you've been on with Biocaptiva. What would it be?
I think that, if I'm honest, I would say I would cast my net wider on where to conduct clinical trials.
Right, okay.
I concentrated on doing them in the UK with the ethos of, if there are problems or things that need address or delivery of bits and pieces or whatever, jump in a car, go solve it.
Yeah.
We don't have the easiest environment. People did suggest other regions to me, and I would have gone back and I would have investigated them more. Right.
Okay. That again is quite insightful. I think you're probably not the only individual I've heard that from.
No, I wouldn't have thought so. Yeah.
I think it's an easy thing to kind of do, though, isn't it? Because you imagine that we've got the NHS, there should be, access and all sorts of good stuff, at one level. But, perhaps the ecosystem is just not as developed as it could or should be, to basically benefit all.
Yeah. Yeah, it's challenging.
Finally, what is the plan for the business going forward? Where are you going to go? What are you going to do?
That's a very good question. The really interesting thing about the business is that, as I say, these polymers have an awful lot of applications. We are a medical business, and of course, one part of it is the medical device business with the Biocaptives. If we can partner with an apheresis company, then we become quite, what's the word I'm looking for? We become quite tight with them on the Biocaptive side, and that's great. That's great. On the polymer coating, I think that's really interesting for the business, because not only are there a lot of applications, but it also means that there's not just the application of, okay, I will license you our polymer, but we can become sort of an outsourced R&D center for coating things as well, because everybody's requirements are slightly different. Even if they technically have the same requirements, it may be that from a business perspective or whatever, they have different requirements. So that's very interesting. And then our polymers, because they're not sample size limited like other extraction methods are, they also lend themselves to environmental issues as well. So you can sample vast quantities of samples. So you can go for looking for all sorts of things in reservoirs or in grey water or whatever, things like that. So that again allows us to have another big interesting part. So it's almost like we have three businesses within the company. And that allows us to go in multiple different directions. When it comes down to it, though, we are a technology company. We provide technology solutions. for collecting DNA. That's what we do. Because of the fact that it's a platform technology, I don't see us doing a lot on marketing our own products, as it were.
So you'll always be in that kind of space, I suppose, as described earlier, very much the cog, a cog in the machine, as it were, that perhaps kind of accelerates other individuals or leverages other individuals.
It's like if you're, for those of us who are old enough to remember, it's like sound by Dolby. It's that sort of thing, and that's great. Again, that is about bringing the maximum possible benefit without technology, and I know that's an important thing, because even from the point of view of aircraft medical, so as you know, we brought to market the world's first fully portable video laryngoscope. You can't believe how excited I was a couple of years ago when I had one of our own products used on me. That was great to see it actually come full circle. If we can get those things out there, you never know. One day it may save my life as well.
Yeah, fabulous. And on that note, Jeremy, thank you very much for your time. That's been very, very interesting and really quite a kind of insightful view into both what you're doing at Biocaptiva, but also, the technology and a kind of wonderful purpose that it serves. And an organisation like that with purpose, there'll be no stopping you.
Thank you. Well, it's always, as you can tell, I'm always excited to share it, because I am very, very passionate about it. So it's been great. Thank you very much indeed.